Most welcome, thankful to the Ethical University and Faculty of Pharmacy for choosing me, giving the lecture on this ATAL FDP program. And also, I would like to congratulations to all the staff members who choosing this topic, green nanotechnology, and the importance in 21st century. Because the older drug systems have a lot of side effects. So now the scientists are looking for the green synthesis of nanotechnology to helpful the patients and reduce the side effects. So that is my primary objective to choose this topic, the green synthesis of chitosan nanoparticles. So chitosan nanoparticles is almost USFDA is approved the chitosan polymer. It's a grass state. It got the grass status now. So in this chitosan polymer have been used for the lot of various drug delivery systems still now. But still the chitosan drug delivery system have some toxic effects. So that you can avoid those toxic effects by using the green synthesis of chitosan nanoparticles. So that way I can choose this topic to deliver the talk on this people to share my knowledge, what we have done the research on the green synthesis of chitosan nanoparticles and the potential application as a drug delivery carrier. So the nanoscience and nanotechnology, it's evolving very fastly now in all the field, especially the pharmaceutical industry. Also now the scientists are looking for nanoscience, nanotechnology to develop the lot of drug molecules and drug delivery system. But still there are a lot of research need to develop the vector or the carrier molecules for therapeutic approach. So that's why the green technology will be helpful to develop the suitable drug delivery system for the suitable drug candidate. So still now that there are a lot of applications. The green synthesis nanoparticles have, like example, the green synthesis nanoparticle developed by using plants and microorganisms. It develops a lot of metallic nanoparticle. The metallic nanoparticle helpful to target the cancer cells without targeting the normal. So that is one of the major advantages of that the green synthesis of nanoparticles. And the green synthesis of metallic nanoparticles have enhanced antimicrobial activity when compared to the ordinary nanoparticles. And photo imaging also, it is possible for the metallic nanoparticles to identify what the stage of the cancer cells. And nanosensor and the nanoparticle can also act as catalyst of some of the reactions. So all the field, the applications of nanoparticle, this nanoparticle may be useful, especially drug delivery. The drug delivery system, the nanoparticles already, a lot of research work have done for develop the target the drug molecule, particular cancer cells or particular cells, particular organs. But still, the drug delivery system may not expose much. So we'll discuss one by one how we are going to address the green synthesis nanoparticles to helpful for the drug delivery carrier. So first, let us see what is the nanomaterials, how the people are developing the nanomaterials. There are two processes. One is top-down approach, another one is bottom-up approach. So top-down approach, nothing but the largest structure of material was broken down into the smaller pieces. So it is also called a physical method. By using milling with small technique they can use that broken down the largest structure. Then the second one is very important one is bottom-up approach. mostly used for developing the nanomaterials. The small atom level will be joined together by using the various plants or biological reactions to make it the larger nanostructures. So the chemical and biological method primarily used for the developing the nanomaterials. You can see here the flow chart. So the synthesis of nanoparticles, there are classified the two, one is top to bottom, bottom to top. So top to bottom, the physical method by mechanical ball milling, thermal laser ablation, pulse wire discharge, so that you can reduce the bigger structure to the bring to the small structure. In chemical also microemulsion methods, phonochemical methods, solvothermal decomposition, you can break the structure. But the bottom approach mainly used for the small atom molecule joint together to develop the bigger nanostructure. The biological method, the green synthesis, there is also called the green synthesis method. So the plant, leaf, flower, fruits, seeds, stems, roots can be used, and microorganisms, bacteria, fungi, algae, yeast, viruses. So all the organism can be used to develop the nanoparticle that is called green synthesis nanoparticle. Why? Because here we are not using any toxic chemicals to develop the nanostructure. Whereas in the physical and chemical method, we are using a lot of chemicals or solvent to develop the nanostructure. So that's only that create a lot of side effects. So recently, the emerging green technology, why means we can avoid the toxic side effect of the drug delivery system. So here you can, in this slide, you can see some of the demerits of the physical and chemical method. So the physical and chemical strategy for the synthesis of nanoparticle had been used, but due to this toxic chemical, it induced a lot of carcinogenicity, toxicity, environmental toxicity, and toxicity problem are quite prominent due to the hazard substance such as reducing agent, organic solvent, stabilizer we are using for developing the nanomaterials. This chemical prevent the agglomeration of lights. The use of toxic solvent and chemical contamination limit the use of nanoparticle in various clinical biomedical applications. So this is one of the major drawback for the chemical and the physical strategies. So this is the reason the people are now looking for the green nanotechnology or biological method has been received much attention for the pharmaceutical industry and pharmaceutical scientists. So now we are going to discuss about how the green synthesis of chitosan nanoparticles. What is the advantage of why you have to choose for the green synthesis of chitosan nanoparticle? Because the chitosan nanoparticle already exists. Lot of research, lot of reports are available. But in recent years, the green synthesis of chitosan nanoparticle received much attention. Why? Because a reliable, clean, biologically appropriate, and environmentally friendly techniques are indeed for the synthesis of nanoparticles. So the biologically appropriate this technique and environment friendly. There is no you're not using for any toxic chemicals. So environmental friendly techniques. The synthesis of nanoparticle using polymer act as a promising candidate due to their ease of processing, solubility, biocompatibility, less toxicity, and also it has the possibility of controlling the growth of resulting nanoparticles. So the chitosan have the main unique characters like the biocompatible one, less toxic one. You can process because it has the amino group and carboxyl group, so you can add any type of functional into the chitosan backbone chain. So the chitosan is more flexible to develop the functionalized drug delivery system. And also one of the main advantage of chitosan is hydrophilic side chain gas. So they play the fundamental role of stabilization and disposition of the nanoparticle, resulting the preventing the agglomeration. So the nanoparticle, one of the main drawback is agglomeration of nanoparticle during the storage conditions. So after some time, the nanoparticle joint together form the lumps. So you cannot redispose easily. But if you are using chitosan, the chitosan will be helpful to preventing the agglomeration. That is one of the major advantages. So these are the unique property the chitosan has. That's why the people are still looking for developing the nanomaterial by using chitosan polymer. So now everyone would like to know, eagerly waiting for how the biological method, what is the basic mechanism, how the biological method can develop the nanomaterial, what is the basic mechanism. So here are some of this, the biomass nothing but either it will be plant or algae or fungi or yeast or bacteria. So how this organism, this plant can helpful to develop the nanoparticle, what is the basic mechanism. So first one is the plant leaves, stems, roots, barks, seeds and all. It have the secondary metabolite. The phytochemical constitution of these plants having the secondary metabolite like alkaloids, flavonoids, sulforins, steroids, tannins. So these chemicals, this phytochemical constitution are helpful to act as a reducing and stabilizing agent of the nanoparticle. So the reducing and stabilizing agent is also capping agents to develop the nanoparticles. So that the nanoparticle will be stable and you can easily prepare the nanoparticle while you are using for the plant extract as herbal extracts. Suppose if you are using some microorganism, the microorganism have like algae. Algae means it has a polysaccharide, has a hydroxyl group, and other functionalities play the important role for the reduction and stabilizing nanoparticles. In the case of the fungi, it reduces the enzyme intracellularly or extracellularly and the procedure of biomimetic mineralization. In case of yeast, the membrane bound as well as systolic oxidative reductase and the quinones can helpful to develop the nanoparticles. In case of bacteria, the microbial cell reduce the metal ion by using the specific reducing enzyme like NADH, dependent reductase and nitrate dependent reductase. So this is the basic mechanism involved in developing the nanoparticle by using the biological method, either plants or microorganisms. So this basic mechanism can be used to develop the eco-friendly or green species of nanoparticles. So the possible mechanism for synthesis of nanoparticle using plant extract. So we are talking about mainly we are talking about the plant extract. What is the possible mechanism? So the still now that the plant extract may be used for developing the nanoparticles metal one, either silver or zinc or like that. So metallic nanoparticles have developed a lot of reports available. So the three important compounds are mainly used. One is reducing agent, stabilizing agent, and solvent of the medium can be used for stabilizing some metal of interest. So based upon the metal which you are copper, zinc, silver. So a lot of metal can be used to develop the nanoparticle by using the plant extract. So the nanoparticle biosynthesis is considered as a green process because the biomass itself can act as a both reducing and stabilizing agent. So this is a very important point. So why that when you're using the plant, it is called as the biosynthesis because the mass itself, the biomass, the extract itself can act as a reducing and stabilizing agents. So we need not to put any extra chemicals to develop the nanoparticles. The herbal extract itself can act as a reducing and stabilizing agent. That's why it is called clean synthesis process. In addition, most of the plant-mediated biosynthesis approaches could be performed in aqueous medium. That is very important. So when you're using herbal extract, you mainly are using maximum aqueous medium, very small quantity of maybe small quantity you can use for the organic solvent. But mainly we are using aqueous medium. Whereas the chemical method, you are using maximum amount of the toxic chemical solvents. So in place of organic solvents, as an additional advantage and apparently more eco-friendly and cost-effective because we are using only for aqueous medium, so we are avoiding all the expensive toxic solvents. So it is also this method can also be cost-effective. So now in this slide, why I'm putting this metallic nanoparticles? So you should understand first how the metallic nanoparticles develop, then only you can understand how chitosan and nanoparticles develop. What is the difference between the developing the metallic nanoparticle and developing the chitosan nanoparticle? So here you can see the metal ions. You go for reductions by using your herbal extract. You directly go for reduction process, reduce the metal ions. So the plant has act as a reducing agent. The plant having the hydrogen group or supplement group, a lot of various functional group that will act as a reducing agents. And it also acts as a stabilizing by capping, so that the nanoparticle, metal nanoparticle can be developed. The metal nanoparticle can be used for the various areas. Now we are going to see about how we can develop the chitosan nanoparticles. And the very important when developing the metallic nanoparticles, there are a lot of factors you have to be very careful. You have to address the lot of various factors. One is the metal salt concentrations is a very important one. The mixing ratio biology extract, metal salt, pH value, temperature, processing period, aeration. So all the factors are very important, crucial one to develop the metal nanoparticles. So you have to optimize it, otherwise you won't get the similar shape or size. The temperature is a very important, playing a major role. It directly controls the aspect ratio, relative amount, size and shape of the nanoparticles. So if you are changing the variation of temperature, the reactive conditions result in change the fine tuning of shape, size, optical properties of nanoparticles. So a lot of external factors involving the developing the metallic nanoparticles. So you have to control all the factors. So here I can put one table for easy understanding. So what are the factors affecting the metal the synthesis of metal nanoparticles? The pH, reactant concentration, reaction time, reaction temperatures. If the pH is, you are not optimized pH properly, then size and shape of the synthesis nanoparticle completely change. The reactant concentrations for not using properly, then shape of the synthesis nanoparticle completely. The reaction time, size and shape of the synthesis nanoparticle completely change. The reaction temperature, if are not maintained properly, then the size, shape, yield, and stability of the synthesis nanoparticles, everything will be affected. So that's why developing the metal nanoparticle by using the plant extract. There are various factors involving, so optimizing the metal nanoparticle process is not so easy. But whereas the polymeric nanoparticle by using herbal extract is so easy one, so there is nothing, external factors may not affect much on the quality of the nanoparticles. That's why that polymeric nanoparticles and the metallic nanoparticles synthesis by using green process is a completely variant. So this mechanism, concept, everything is different, and polymeric nanoparticle developing by using green nanotechnology is a completely different one. So now we are focusing on the chitosan nanoparticles. So why I am choosing the chitosan? The chitosan possesses the free amino and hydroxyl group that enable cross-linking. So you can easily cross-link the polymer by using because it has the free amino and hydroxyl group. In a lot of reports available, they are using various cross-linkers to develop the chitosan nanoparticles like formaldehyde, tripolyphosphate, glutaraldehyde. But these all the cross-linkers have the toxic effect. And the structure of the nanoparticles is not rigid. The mechanical strength of the nanoparticles is not appropriate. And sometimes some of the chemicals may not be non-biodegradable property. Thus, it is essential to find a non-hazardous, biodegradable, and effective cross-linker for the preparation of chitosan with better mechanical properties. So the existing method having a lot of demerits while developing the chitosan nanoparticles. So that's really the green synthesis of chitosan nanoparticle nowadays has received much attention in the pharmaceutical industry and pharma scientists. So because the green synthesis nanoparticle have the better mechanical strength and there is no side effect and biodegradable. So the green synthesis nanoparticles here So in this study, I'm using the cinnamon is a herbal. From the cinnamon, you can extract the cinnamaldehyde. The cinnamaldehyde has the property to cross-link the chitosan. It directly binds the primary amino group of chitosan backside and it forms the cross-linking of chitosan nanoparticles, form and use the chitosan nanoparticles. So this basic concept I'm using to develop the green synthesis of chitosan nanoparticles. If you're using the cinnamaldehyde, it is a herbal one, it is a non-toxic one. It is also have some antibacterial property and functional properties. Since it has an aromatic conjugation and an aldehyde group, it was envisaged that the cinnamaldehyde may be used as a cross-linker for the preparation of chitosan nanoparticles. So this is a basic hypothetical idea to that a lot of scientists are now recently some of the scientists have used cinnamaldehyde as a cross-linking agents for green synthesis of chitosan nanoparticles. Because this nanoparticle have the non-toxic and biodegradable and good mechanical strength when compared to existing nanoparticles. So here I can put on schematic diagram for each understanding how the chitosan nanoparticle, the green synthesis chitosan nanoparticle have developed. Very simple process. First, you have to take the chitosan and make it into the solution by using water. Then you can take the cinnamon bark and go for extraction process. And you can extract the cinnamaldehyde from the cinnamon bark. When you're mixing the both cinnamon extract and the chitosan solution, the cinnamaldehyde will be completely bind with the primary amino group of chitosan. It forms cross-link and it develop the chitosan nanoparticles. That is called final product imines base. So you can see the diagram, so you can easily understand so how the chitosan will be there. The cinnamaldehyde will be completely cross-linked, so the chitosan nanoparticle develop. Very simple process. So it is easy to scale up, easily reproducible, and there is no toxic effect. So that's only the use this chitosan nanoparticle nowadays have received much attention from pharmaceuticals. Now this chitosan, the green synthesis chitosan nanoparticle can be used as a drug delivery carrier. So still now they develop only antibacterial, antimicrobial activity of chitosan. The enhance is already proved that a lot of reports are available. But the green synthesis nanoparticle can be used as a drug delivery carrier. So here we are using the insulin. You are taking the insulin as a protein replacement drugs. So still the insulin, the developing the oral bioavailability of insulin, enhancing the oral bioavailability of insulin still is a challenging question for the pharmaceutical industry, pharma scientists. So we are using the we are trying to develop the green synthesis chitosan nanoparticle for insulin, so that the green synthesis nanoparticle can enhance the bioavailability of insulin. It can be helpful for the patient for treating the chronic disease diabetes mellitus. So already know about what is the diabetes mellitus and how it affects the human populations around the globe. So it is one of the very familiar disease to affect the most of the person, 50% of the populations as per the WHO report. So the insulin is one of the therapy to give the diabetes patients for chronic diabetic patients. But the injection is still now the insulin is given for the supplementary injections only. So a lot of clinical trials, lot of what you can say, a lot of clinical trials, a lot of companies looking for developing the oral delivery of protein peptides insulin. So there are a lot of various approaches that developed or reported still now. So here we are in our group, we are just trying to develop the improving the insulin bioavailability by using green synthesis chitosan nanoparticles. So very simple process. So the insulin loading the nanoparticles is performed a passive absorption technique. Because the insulin is so sensitive one, so you cannot simply add it while preparing the developed nanoparticle. So after the development of after the filtering, after the drying of the nanoparticles, you can put into this insulin with the nanoparticles. So through the passive absorption technique, the insulin can enter into the green synthesis chitosan nanoparticles. So the maximum amount of the chitosan having for large the green synthesis having a large surface area. So it absorbs the most, the passive absorb through the absorb maximum amount of the insulin. So how much amount of insulin absorbed enter into the nanoparticles, you can easily determine by using the microbial technique. So the drug loading and loading efficiency and the loading capacity of the nanoparticles was determined by using the following equations. And we found that the 0.5% of insulin having for the maximum loading capacity, loading efficiency. If I increase the concentration, there are some much changes in about the loading efficiency, loading capacity. So 0.5% of insulin is the optimum concentration. So why you are choosing this technique? Because of the protein updates, the drug insulin should not expose the horse process. If you're exposing the horse process, the insulin stability will be decreased. So that's why you are using the passive absorption technique. So the loading efficiency, loading capacity of the nanoparticle, different punches loading was significantly different, so that we are using a 0.5% of insulin loading was selected as optimal concentrations. So these are the beautiful image of the green synthesis chitosan nanoparticles. You can see how it the spherical and uniform distributions. When compared to the existing process, the green synthesis nanoparticle having for mechanical strength is good and the uniform distribution also possible. It is clearly evident that same image shows that the nanoparticle having the spherical and a good mechanical strength. So the nanoparticle is spherical in shape with a smooth surface. It can be observed at polymer cross-link with cinnamaldehyde was successful. The cinnamaldehyde can completely cross-link and the cross-link of the chitosan, so that the mechanical strength of the chitosan film will be enhanced, which lead the uniform formation of the nanoparticles. The free amino group of chitosan is cross-linked with the aldehyde group of cinnamaldehyde to form chip base. That is the basic mechanism how the cinnamaldehyde and chitosan develop or lead to the nanoparticles. So the free amino group of chitosan is cross-linked with the aldehyde group of the cinnamaldehyde to form the chip base. Then we observe that the zeta potential of this nanoparticle is a positive one. That is a very good sign, because there are the primary amino, the unreacted primary amino group responsible for the positive zeta potential. So the positive zeta potential can easily interact with our cell membrane. So the tight junction can be easily opened, so the membrane permeability may be enhanced. The particle size also was found by using the laser diffraction laser technique analysis. So the particles are found with the nano size, and the PD value is very less than 0.3. So it indicates the uniform distribution of the particles. So now one of the main advantage of the green synthesis chitosan nanoparticle when compared to existing method. Next, we have conducted the gel electrophore system for identifying whether after the encapsulation of the insulin, and the insulin will be active or not. How we are going to check? Insulin will be there, but the activity of the insulin will be reduced or it will be stable. We have to found by using the gel electrophoresis. So here we can found that the insulin will be stable and it's active one inside the microspace. When compared to the pure insulin, so the molecular weight and the activity of the insulin will be same. So this technique will be the insulin will be the stable one inside the microspace. Can prove that. So insulins are stable after encapsulating into the chitosan nanoparticles. Next, we perform the in vitro release of insulin release from the green synthesis chitosan nanoparticles by using the two different pH and gastric pH and the 7.4. It release very minimum quantity of drug from the release at the gastric pH because we have already found that the chitosan cannot release the drug at the gastric pH. After the cross-linking, it's the mechanical strength is good. So only 2 hours it will release only for 20% of drug will release, 20% of insulin will release. Once the chitosan nanoparticle, green nanoparticle put into the pH 7.4, then it starts swelling and it slowly releases the drug. So we observed that the results lead to the assumption that in vitro release of insulin seems to be continuous dependent upon the times. And the amount of insulin release gradually increases with the time of nanoparticles. And the observed release pattern indicates the diffusion-based mechanism. Because the chitosan is a rigid, the cross-linked with cinnamaldehyde is a mechanical sense simple rigid one, so it is slowly getting hydrated with the pH 7.4 and it is swell it and it is slowly the diffusion pathway will be created through the diffusion pathway and the insulin will be released from the nanoparticles. So for finding the diffusion mechanism of the green synthesis chitosan nanoparticles, we are applying the Pepper's model equations. We'll plot the graph log of time versus log of cumulative drug release. That n values found at 0.46 with the good correlation coefficient of R square value 0.9. So this value indicate that the release from the insulin from the nanoparticle following for frequent diffusion method. So frequent diffusion is the predominant mechanism where the insulin release from the nanoparticles. Next, we have performed the in vivo studies. You can see how the blood glucose level, plasma glucose level, and how long the plasma glucose level control and how magnitude the plasma glucose level control. Everything are checked by using the stabilization induced animal model. So where first we inject the stabilizations to the rats and induce the plasma glucose level with more than normal one, 280 to 380 milligram deciliter. That rats will be called as diabetic rats. The rat was chosen for the treatment. Like one is first one is we are dividing the 4 groups. The first group is a control. The second group is support insulin solution, 200 per kg. The oral insulin we are giving and also found the comparing how best our nanoparticle can improve the bioavailability of the insulin. So we can see after the dose administration, after the nanoparticle administered to the rats, we collect the blood sample and find out the glucose level, and we found out the relative pharmacological efficacy. It is called area under Yaboka. By using the glucose level, we plot the graph. We found that 3 full times the relative pharmacal efficacy are increased while using the green synthesis nanoparticle when compared to oral insulin. And also we have collected the plasma sample from the plasma sample. We'll find out the glucose level. We determine the glucose level by using HPLC method. And we can calculate the various pharmacokinetic parameters, Cmax, Tmax, AUC, and relative bioavailability. The oral insulin, the Cmax is very, very low, significantly low when compared to supplement injections and the green nanoparticle. So the relative bioavailability of the insulin will be enhanced 3-fold times while treating the group with the green synthesis chitosan nanoparticles. So you can see that the chitosan nanoparticle to control the plasma glucose level for a longer period of time when compared to oral insulin supported injection. This is one of the major beneficial for the green synthesis chitosan nanoparticle as a delivery carrier of the insulin, oral insulin. And also that you can see this graph that amount of plasma insulin available in the insulin available in the plasma is significantly higher when compared to supported injection. So the AUC value of the insulin will be for after administration of the chitosan nanoparticle very significantly higher than supported insulin injection. So the bioavailability of the insulin may be improved by using the green synthesis chitosan nanoparticles. It is clearly evident that from this pharmacokinetic parameter, the results reveal that the green synthesis chitosan nanoparticle enhance the bioavailability of insulin, oral insulin. The plasma insulin concentrations, the results was clearly evidence that the ability of chitosan nanoparticle to enhance the absorption of insulin in the gastrointestinal tract. So insulin is the high molecular weight protein drugs. So it is very difficult to permeate the skin. But if you are using as a chitosan as a carrier, then the chitosan may helpful to permeability of the insulin because it has a positive charge. The positive charge may be helpful to, and also the enhanced surface area with a high number of available free amino group to intact that is responsible for the positive charge of the nanoparticles. So this positive charge will be easily intact with the negative side of the tight junction of the epithelial cells. Through that drug molecule, the insulin can be and widening the paracellular transport route so that the insulin can easily penetrate enter into the plasma. So this is the main mechanism. The oral bioavailability of insulin may improve by using green synthesis of chitosan nanoparticles. The existing nanoparticle having for the small surface area, so that may not be enhance the bioavailability this much level. But if you are using the green synthesis of chitosan nanoparticle having for the enhanced surface area and the excess amount of the free amino group is available, so that the positive charge is very high, the positive charge intact with the negative charge of the epithelial junctions. So it will be the widening of the paracellular route so that the insulin can easily penetrate the membrane and reach the plasma, blood circulations. So we already told that the relative pharmacological efficacy of the chitosan nanoparticles almost threefold higher than the oral insert administration. So this is revealed that the chitosan nanoparticle enhance the bioavailability of oral insulin. And finally, we have conducted the stability studies. Green synthesis chitosan nanoparticles. From the stability report, we can found that there was no change in the parameter of entanglement efficiency or particle size. There is no agglomeration, and the in vitro release also same initial and final. So, thus from the optimum results, the green synthesis nanoparticles can be considered stable under tested conditions. So, stability wise, the green synthesis nanoparticle is good when compared to existing chitosan nanoparticles. And also, we have conducted some cytotoxic studies. Here we didn't show the report, but we have conducted the cytotoxic studies for the using for different cell lines. Cytotoxic effects also very less, so that should be very safe for the green synthesis nanoparticles. And also, we have performed some histopathology studies. So from the results, we can found that the green synthesis nanoparticles very safer one. There is no toxic effects. So from the above data, we have concluded that the green synthesis nanoparticles are able to reduce the glucose levels for a longer time and increase the overall viability of insulin. This observation indicates that the green synthesis of chitosan nanoparticle has a potential to serve as a oral insulin carrier. So this is one of the, when compared to the existing chitosan nanoparticle, the green synthesis nanoparticle has the potential to serve as a oral insulin carrier because it has a high surface area and it's a natural one, less toxic, and mechanical state is good, and uniform size distributions. So a lot of advantages having when compared to the existing chitosan nanoparticles is prepared by using glutaraldehyde and TPP triphosphates. According to results, the new carrier system described herein may act as a promising tool for the oral administration of insulin and is similar for various protein and peptide drugs. So this green synthesis nanoparticle not only for insulin, we can use for some other protein peptide drugs which cannot the poor bioavailability because of the high molecular weight. So this green synthesis nanoparticle may enhance the permeability of the protein peptide drugs. It is a hydrophilic one, so it can be easily can cross the membrane. So by using the green synthesis nanoparticle, we can achieve that, enhance the permeability and enhance the permeability of the drug is a molecule having for the poor permeability in the CIT per membrane. So from this study, we can conclude that the advantage of green synthesis nanoparticle when compared with the existing one, the green synthesis nanoparticle is free from toxic chemical. So in the preparation process of nanoparticle, we are not used any toxic chemicals here. Glutaraldehyde, triphosphates, glutaraldehyde is toxic one, triphosphates is also toxic one. So we are using for the natural cross linker, the cinnamaldehyde. It obtained from the natural resources. It is also now the food supplement. So it is we have taken daily for cinnamaldehyde small quantity. So it will be very biocompatible with our immune system. And next, the important quality is the uniform particle distributions. So because the cinnamaldehyde is cross-linked with the primary amino group of chitosan, so the size of the particles will be the uniform. Through the interaction between the cinnamaldehyde and the amino group, the process will be very slower one, but is It is a rigid one. It will each and every molecule crossing each other, so it will forming the uniform size. Whereas if you are using glutaraldehyde or TPP, immediately it forms the crosslink. So that only the uniform size may not be achieved over there. But here the crossing time was more, but it will be a uniform one. And the crossing was very well with the primary amino group and the cinnamaldehyde group. So the structure, the rigid spherical structure will form. So it can be used for it can control the drug release for longer period of time. So we can use as a drug release system for various drug molecule, so that the drug release will be very slow when in the plasma drug concentration for longer period of time. So we can each achieve the steady state plasma drug concentrations for longer period of time. So if you are maintaining the steady state plasma concentration for longer period of time, that the disease will be controlled for longer period of time. It may be improve the patient compliance. So that is our very primary objective, to develop the new delivery to enhance the patient compliance. So this type of the green synthesis chitosan nanoparticle may be helpful to improve the patient compliance. One is by reducing the side effects, toxic effects, and one is the prolonged effect. And also we can, the burst release also is not there here because of this structure. It will be slow, so it will be hydrated really slowly. So there is no burst release from the nanoparticles. And the increased surface area. So it has, when compared to existing nanoparticle, this green synthesis nanoparticle have for the increased surface area. This surface area will be helpful to release the drug molecule very slow manner. And also just the amount of drug release also will be reach the minimum plasma effective concentrations very fastly. So this type of nanoparticle can maintain the plasma drug concentration between therapeutic window. It means minimum effective, above the minimum effective concentrations and below the maximum safety concentration. So that green synthesis chitosan nanoparticles helpful to maintain the concentrations with the therapeutic window. So it will improve the patient's compliance. And it is prepared from all the materials are used to prepare the chitosan nanoparticles. Nanoparticles a natural one, so it is available very easily, and the process is a very simple one, so it is very cost effective. And the process is a very simple one, so you can easily scale up. In the pharmaceutical industry, they are always in the laboratory. We can develop the lot of nanoparticles, but when you go for the scale up, the lot of problems they're facing. But this process will be very simple one, so the scale up will be very easy. And the factors affecting this nanoparticle is very limited one, so you can easily optimize by using various techniques. Nowadays the QBD will be available, so while developing this type of nanoparticle by following the QBD, so you can optimize very fastly, and the batch-to-batch reproducibility will be ensured. Here I'm not showing that the QBD results because we have the data, but there is a time is not sufficient to develop the QBD process. So that's the result I didn't share with you. If I have time, I'll share the QBD data also in future. Based on the stabilities, we can found that the dosage forms, the agglomeration is not there during the stability period. One of the major problem with the nanoparticles is agglomeration during the storage period. But the green synthesis chitosan nanoparticle, there is no, we can found any agglomeration. So this type of nanoparticle can claim that's a good stable one. And the entire efficiency of the insulin also cannot be changed. And the unit release profile also will be same for initial and final. So the initial release profile and the amount of drug available in the drug content and agglomeration. The three are the major problem for nanoparticle, but three problems can be solved by using this green synthesis chitosan nanoparticles. So these are all main advantage of green synthesis nanoparticle when compared to existing one. So that's only we are go for depth analysis, depth research, depth investigation of this green-size nanoparticle using various drug molecules in future, and also we'll try to develop green-size nanoparticle, go for patent and go for clinical trials also. So with this data, I'll end up my talk. So if you have in future, if you have any time, we can discuss regarding the QBD analysis and the remaining how and what are the other data we have, we can share with you. So finally, we can conclude that this way. So the green synthesis of polymeric nanoparticle, either we can use chitosan or some other polymer to develop the nanoparticle. That nanoparticle may have good, stable, and good control the drug release when compared to existing one. So I urge all the scientists who are present here. So try to develop the dosage for the drug release system by using the green synthesis technology. It may be helpful to improve the quality of your nanoparticles. So these are some references. We have gone through some of the references to develop this technology, to develop this theoretical concept. The references may be helpful for us to carry in the research. So now we have enough time to discuss